Okay, we're talking about enzymes in section 3 for chapter 3. And with enzymes, in our learning objectives, we want to be able to describe the general function of enzymes, describe the key structural components of enzymes, identify the places of the body where enzymes may be found, explain the steps by which an enzyme catalyzes a reaction, describe cofactors and their role in reactions, and identify the 6 major classes of enzymes and the general functions of enzymes in each class, describe the naming conventions for enzymes, describe how or define how enzyme and substrate concentration affects reaction rates, and explain the effects of temperature on enzymes, while also being able to describe how pH changes affect enzymes. And describing how competitive and non-competitive inhibitors control enzyme action, distinguishing between a metabolic pathway and a multi-enzyme complex, explain the role of negative feedback in enzyme regulation, and identify and explain the processes involving phosphate that commonly are used to regulate enzymes. And if you can do all those things, you're going to be quite an expert on enzymes. That being said, you should relatively be able to do that at least fairly well without too much reference if you've gone through this information a couple times. It'll require a couple times to get all that information, but enzymes are pretty important, which is why there are so many learning objectives here when it comes to enzymes. Enzymes, their function, they're catalysts, right? They accelerate normal physiologic activities. They decrease the activation energy required for cellular reactions. N-catalyzed, there's no enzyme present, and catalyzed, there's an enzyme present. They only facilitate reactions that would already occur. So those reactions will occur whether or not there are enzymes. However, when the enzyme is present, it's going to dramatically increase the rate of product formation, possibly as much as 10 billion times, which is insane, but possible in some situations with some enzymes. That said, the activation energy, EA, it's an exergonic reaction we're talking about. In this case, we're talking about energy supplied here. We're starting with the reactant, which is sucrose, and the active energy to basically to release over the course of reaction for breaking apart sucrose into glucose and fructose is going to take, if we're not having an exact amount here, this much, right? With the help of the enzyme, it dramatically reduces the activation energy required to break down sucrose, as far as then creating and then break it down into glucose and fructose. That's a monstrous difference between not having the enzyme and having the enzyme. From a general perspective, reactions will occur with or without enzymes, but of course, it dramatically enhances the metabolic capacity of our body, energy conservation, increased efficiency when we have the benefit of enzymes present as well. With enzyme structure and location, most enzymes are globular proteins. They range in size from small, about 60 amino acids, to large, about 2,500 amino acids. They have a unique three-dimensional structure in the protein chain called their active site. Okay? They temporarily form what is known as an enzyme substrate complex. So in this picture, that active site specificity, each enzyme is very specific to what substrate it will work with. It permits only a single substrate to bind, and that's the only kind it will allow for any particular enzyme. Okay, and this helps catalyze only one specific reaction. And we'll get a clear picture of this shortly, kind of how this looks from the standpoint of the reaction as a whole. We'll come back to that. There'll be a picture later. The location of enzymes, some are within the cells, like DNA polymerase, which helps form new DNA. Some become embedded in plasma membrane, like lactase in the walls of the small intestine, which helps digest lactose. Right, lactose, not much of a stretch there. Just like DNA polymerase, you can figure out that helps break down the polymer, which is DNA, right? A lot of the enzyme names are given away by their name itself, what their function is going to be, generally speaking, or at least fairly easy to infer, right? Some are secreted from the cell, like pancreatic amylase, which is released from the pancreas to participate in starch digestion, okay? As far as enzyme catalysis, substrate enters the active site, forming an enzyme substrate complex. The enzyme changes shape a little bit, which results in something called the induced fit model. And this change in enzyme shape stresses the chemical bond, permitting new bonds to be formed, and the products are released. Then enzyme may repeat the process. So here we have a graphic representation of a decomposition reaction involving an enzyme. In this case, we have lactose as the substrate and the enzyme lactase to help break that down in this decomposition reaction. So the substrate lactose binds to the enzyme, forming this enzyme substrate complex. Okay, the enzyme changes shape, which is resulting in this induced fit between the two, the substrate and the enzyme. And the bonds then broken between glucose and galactose. So now you have glucose and galactose, and you have this free now enzyme which is able to bind and begin the process anew. In a synthesis reaction, we have the substrates glucose here, two different monomers, glucose monomers, and then we have glycogen synthetase, which you can infer glycogen synthetase, synthesizing glucose into glycogen, right? So this glucose substrate binds to the enzyme, forms this enzyme substrate complex. The enzyme changes the shape once again, resulting in an induced fit between the substrate and the enzyme. The bond's formed, forming glycogen, and that glycogen is going to be basically stored in muscles or liver, okay? And then the product is the glycogen is released, and then the enzyme is free to bind with other substrates once again, but only with glucose for the glycogen synthetase to formulate glycogen, okay? And going back to the previous slide, when you got glucose and galactose free here, generally speaking, when you have free basically similar monosaccharides, simpler carbohydrates, the probability is it's going to need to be used up immediately by the body. It's not going to be able to store it easily. There's a common thinking sometimes that people think when there's sugar present, it's going to be able to bind pretty easily and form glycogen. That's only if there's glycogen already present in the system and the body is getting what it needs in this moment. Otherwise, it's going to try to use it up. If it doesn't have stored glycogen, it's going to try to use it up. And of course, that's going to create all sorts of other responses as well, which may or may not be ideal for the individual at that given moment in time. It's never it's good or it's bad. It's never. It always depends on so many other factors, right? These are just some basic factors we're going over right now. And then try to figure that out in later units. We'll try to tie this all together, probably more in AP2. The cofactors are basically molecules or helper ions that are required to ensure that a reaction occurs. There's a non-protein organic or inorganic structure, inorganic factors, cofactors attached to an enzyme, like the zinc ion required for carbonic anhydrase to function. Organic cofactors called coenzymes, like vitamins. Vitamins are coenzymes, or a modified nucleotide serving as coenzymes. And there's also these enzymes, 6 of them, 6 categories here, major functional classes. Oxidoreductase, which are enzymes in this class that participate in oxidation reduction reactions, okay, like dehydrogenase. Transferase, all these enzymes basically are within chemical structures like kinase, hydrolase, isomerase, ligase, and lyase, okay. Fortunately for you, you're not going to know every nuance of all of those, but you want to know, generally speaking, which functional categories exist. The enzyme names based on typically the name of the substrate of the product, right? It's subclass suffix ase, right? Pyruvate dehydrogenase transfers hydrogen from pyruvate. You can kind of infer that there. Then again, DNA polymerase helps form DNA, and lactase helps digest lactose. Okay. The effect of the enzyme and the substrate concentration rate of a chemical reaction can be accelerated by an increase in enzyme concentration or an increase in substrate concentration. Okay? And it increases only to the point of saturation. It comes to a saturation point when it can't continue to increase, right? When saturation is reached, that all enzyme molecules are engaged in a reaction, it can no further speed up that reaction. The reaction cannot increase any further than it already is. That case, we're talking about the rate of reaction here, relative to the concentration of substrate. Either one goes up until a point of saturation. That levels off, whereby you can no longer speed up the rate of reaction. Okay, very general, but important to understand. The effective temperature. The three-dimensional shape of an enzyme is dependent upon the temperature. Okay. Human enzymes function best at optimal temperature around 98.6 degrees Fahrenheit, around, like we mentioned in a previous unit, not exactly 98.6, but close, within a range to that exact temperature. A moderate fever results in more efficient enzyme activity, meaning enzymes are going to pick up, they're going to be a little bit of that increase in heat. Basically, a slight change of pH might increase the activity a little bit. It may be efficient from the standpoint of them being able to do their job. Moderate fever meaning activity is going to increase your body heat, right? Moderate, meaning that's barely, like, not even a degree. Okay, very, very small change. Severe increase in temperature, like a couple degrees and more, let alone technically about 105 degrees or more, is really where 105 or more is where we really talk about the probability or the increased likelihood of protein denaturation with a loss of function because of the unraveling of those proteins. The effective temperature on enzyme activity. We have this three-dimensional shape of the protein. It's more rigid at cooler temperatures, right? As the temperature rises, optimum temperature for maximum protein flexibility of intact human enzymes around 40 degrees Celsius. We're talking Celsius here. And then enzyme denatures as weak intermolecular interactions are broken at higher temperatures beyond that. Okay, right about that point, that's when you start seeing denaturing after that. So that's why with activity, exercise with an increase in temperature to some extent, mild or moderate, I'd call it mild, actually is going to be more efficient, make a more efficient system in the short term. We cannot have a higher temperature than 98.6 for a lengthy period of time. This might give you the idea that somehow we could actually increase temperature more than normal for long periods, and that would not be ideal. For short periods, it can actually increase the efficiency of enzymatic activity or as catalysts when we exert, right, when we move, increase body temperature. Effective pH, enzymes function best at optimal pH between 6 and 8 for most enzymes, slightly acid or slightly base, right, towards 6, slightly acid, or toward 8, slightly base. Changes in the hydrogen disrupt electrostatic interactions, right, and the more higher presence of this, more acidic it becomes, lowering that pH. The enzyme will then lose shape, but technically high base too high as well can absolutely cause the same denaturation issue. But that being said, typically referring more toward acid with excess hydrogen ions. Optimal pH may differ to some extent. Enzymes working the lower pH of the stomach are examples of that can vary a little bit from this range. Effects of pH on enzyme activity. So as the enzyme denatures with excess hydrogen, or basically with a basically lost too much hydrogen, or more hydrogen, more acidic, or too little hydrogen, too base, it's going to denature either way. Okay, and the optimal pH, of course, is neutral at 7, but or between 6 and 8, we really should say, that bell curve, right? As far as inhibitors, they bind enzymes and prevent enzymatic catalysis. They prevent overproduction of product. Some's got to regulate the enzymes, right? Technically, DNA is really what helps regulate enzymes. Later release of inhibitor allows it to function, and catalysis continues. Inhibitors can be competitive or non-competitive. A competitive inhibitor resembles a substrate and binds to the active site of the enzyme. Okay, it competes for occupation of an active site with other similar enzymes, and with greater substrate, there's less likely the competitive inhibitor will occupy the site, right? With greater substrate into the system, the enzymes don't need to, the competitive inhibitor does not need to be so competitive, if you will, right? With less substrate, more likely inhibitor will occupy the site, and then you'll have competitive inhibitors that are looking for sites to bind with. Non-competitive inhibitor, it doesn't resemble the substrate, okay? It binds a site other than the site, also called the allosteric site. It induces conformational change to the enzyme active site. It's also called an allosteric inhibitor. Okay, it's not influenced by the concentration of the substrate at all. So whereas the competitive inhibitor is absolutely affected by the concentration of substrate, the non-competitive inhibitor is not affected by the concentration of substrate at all. Multiple enzymes usually required to convert initial substrate to final product. Like one enzyme is not going to solve all, right? In a metabolic pathway, it's a series of enzymes that are playing a role. The product of one enzyme becomes a substrate of the next with chemical breakdown of glucose. We already saw that cycle before. In a multi-enzyme complex, it's a group of attached enzymes work in a sequence of reactions like the pyruvate dehydrogenase. And that is our section for enzymes.